Hi, good afternoon. Welcome to everybody who joined today's webinar. So today's webinar is about fighting COVID-19. And in this three-part webinar series, we will discuss how biomarker discoveries, also the new therapeutics and treatments are developed to fight these global pandemics. Before we start today's webinars, I had a few housekeeping tasks for everyone. So if you have any questions regarding the topics and you want to ask our speakers, please type in your questions in the Q&A section. Alternatively, you can also interact with other audience and also our speakers in the chat sections. So we know that until today, the COVID-19 pandemic has caused about infected case of more than 50 million around the world. And there is also a global fatality of more than 1.2 million. As you can see from the map here that North America and also Europe are currently having a daily cases more than 10K or even 20K. And we are also facing a second wave around the world in different countries, including Europe, Asia. Now, how to fight these global pandemics and how do we, not just for the vaccines, but also for the treatments and prevention from the next virus. On NanoString, as a leader in life science research tool, we are committed to fight this pandemic together with our researchers. So in the last couple of months, NanoString had developed new gene expression tools where you can profile more than 700 human genes and 50 pathways in immunology and also infectious disease, allowing our researchers to identify signatures which are related to host response dynamic and also disease severity. We also developed spatial biology tools where we allow researchers to utilize our genomics DSP platforms to look into tissues of COVID-19 infected patients. For example, the lung tissues, for example, the heart tissues or liver tissues, where virus interact with the host immune cells. Now, in today's session later, you will hear some very exciting stories from our speakers about how spatial resolved information can aid in the biomarker discovery. Without further delays, I also want to go into introduce you the three parts of this webinar. This is only the first part of our webinar series. And in the next week and also the week after, we also had two more sessions talking about the new therapeutics and also the next wave and next threats, where we focus on topics on how to prevent the next pandemics. And this three-part webinar series is co-organized by NanoString and also National Taiwan University. Now I would like to pass the opening to Professor Ruby Huang from National Taiwan University. Professor Huang is also the main organizer for this three-part webinar series. Prof. Huang is an expert in ovarian cancer, but she also had a high interest in spatial biology as well as the COVID-19 research. Prof. Huang is the first one to introduce the genomics DSP platform in Taiwan. And currently, she's also having an international collaborations with researchers across Asia Pacific on COVID-19. So Prof. Huang, I would like to pass the introduction to you. Thank you.
Thank you, QQ. Thank you everyone for joining this webinar series jointly organized by National Taiwan University and NanoString. The year 2020 has been very challenging for everyone facing the COVID-19 pandemic. Being the flagship leading university in Taiwan, National Taiwan University has formed a research center for epidemic prevention science to dive deep into the scientific discovery for emerging infectious diseases. So this webinar series is a showcase that we want to deliver to the community of what NTU is currently pursuing in terms of the COVID-19 research, and share with the research community of what we have achieved and what we can, achieve further, bringing together the research community for cross-border collaboration. So once again, I'm very excited for this joint webinar series with NanoString. Thank you very much joining us. And we have a fantastic two speakers for today's webinar, and I hope you will enjoy their talks. Back to you.
Thank you, Prof. Huang. So next, I would like to introduce our first speakers, Prof. Chang Lee from National Taiwan University. So Prof. Lee today, he will discuss about the role of how biomarker in diagnosis, risk stratification, and also monitoring of COVID-19 patients. And this is a very interesting talk. And then if you have any questions, please feel free to raise these questions in the Q&A session. And if time allows, we will address this question with Prof. Lee after his sessions. So Prof. Lee, I will start your sessions later. And before that, we also want to introduce another speakers. So today, Dr. Arutha Kulasinghe from Queensland University of Technology will also share his research work on COVID-19 research. Dr. Arutha Kulasinghe, he is an expert in both cancer and also infectious disease, particularly in his recent research for COVID-19. He used spatial biology to uncover prognostic marker, which may impact host response and gene signatures. So without further delays, I would like to invite Prof. Lee to start his session, and that will be followed by Dr. Arutha Kulasinghe's sessions. Hi, Prof. Lee. You can start sharing your slides now.
Yeah. Hi. First of all, I think Ms. Tan for the introduction, and thanks for Professor Huang that organized this so meaningful event. Today, I'm going to share my experience and summarize some of the current evidence of the biomarker test in COVID-19. Excuse me. Sorry. Okay. As mentioned by Ms. Tan, currently there's already about 50.7 million.
Prof. Lee, so just had a quick interruption. So the slide is not showing yet.
Okay. So
yeah.
So can you see the slide now?
Yes, we can see the slide now. Thank you.
Okay. Sorry for a little interruption. Okay, so first of all, let's start with the current situation of the COVID-19. This is the latest update that there's already 50.7 million of cases of the COVID-19. And death mortality cases can be total to 1.2 million. And what makes things worse, you can see that the trend actually is accelerated. So we do not peak yet. So this is a very terrible situations, what we are facing now. And if you see the world map, you can see that the Europe and Russia and North and South Americas are areas that are affected by the COVID-19, most severe areas. And this is currently drug trackers by the New York Times. Currently, you can see there's only one drug that is approved by the FDA, which is remdesivir. And two interventions that is widely used. One is a prone position, the other is mechanical ventilations. So obviously, currently we have very, very rare weapons or treatment approaches toward the COVID-19, except for the remdesivir. Most of them actually is the supportive care. So we need to understand the pathophysiology of the COVID-19 before we can really develop a very effective immunotherapy. And for the remdesivir, 2 months ago, we published analysis with Professor L.J. Wei from the Harvard Universities. In the NEXAM trial, you can see that if we analyze mortality as the endpoint for remdesivir, actually it did not improve the mortalities in the clinical trial. However, Professor Wei proposed that the analysis maybe wrong, because death is a competing risk for recovery. So we use another way to reanalyze the data, and we show if we use the recovery as the endpoint, actually remdesivir has better performance for recovery than the placebo. So the average disease-free days during the observation period, you can see that for the remdesivir, it's 14.1 days, but for the placebo, it's only 11.9 days. Although remdesivir did not improve the mortality in the clinical trial. And the other hope is reside on the vaccine, but currently there's only 11 vaccines on the phase 3. Yesterday, the good news from Pfizer is really an exciting news. And also Moderna is one of the vaccine that is already complete the recruitment of the phase 3 30,000 patients. So we may reasonably hope that the vaccine will be an effective preventive measures early next year. So let's start with the pathophysiology of COVID-19. We already know that the SARS-CoV-2 virus enter our human body or human cells via the ACE2 protein. And the ACE2 actually, you see, is an important mediators that can convert angiotensin 1 or angiotensin 2 to other form of angiotensin that can avoid angiotensin going to the pro-inflammatory pathways. We can summarize the inflammatory effect of angiotensin using this diagram. If the angiotensin be converted by angiotensin converting enzyme to angiotensin 2 and coupled with the AT1R, that's the angiotensin 1 receptors, it will induce the subsequent pro-inflammatory reactions. However, if this angiotensin are converting by the ASE angioconverting enzyme 2 before they couple with the AT1R, the net effect will be the anti-inflammatory effect. So it is, and we know that the ACE2 is distributed in different type of organs across the bodies. This can explain the multi-organ dysfunctions of SARS-CoV-2 in severe cases. So let's go back to this slide. We know that if we can use the ACE inhibitor or ARBs that block the pathways of angiotensin to the pro-inflammatory pathways, we probably can alleviate the inflammatory cytokine storm induced by the SARS-CoV-2 virus. So it is really interesting whether ACE inhibitor or ARBs, the two commonly used antihypertensive medications, can be used as a immunotherapy for the COVID-19. Before such trial is available, actually, we did an population-based research based on our National Health Insurance database research last year. We analyzed whether the renin angiotensin aldosterone system inhibitors could improve the short-term mortality of sepsis. And after comprehensive analysis and control for the confoundings and several sensitivity analysis, we conclude that prior use or pre-admission use of ACE inhibitor or ARB are associated with the reduced 30-day or 90-day mortality rate. So based on our analysis, we think it is a reasonable interventions to use renin angiotensin aldosterone system inhibitors to improve the immune response of SARS-CoV-2. Okay, and then let's go to the biomarkers for COVID-19. For clinically definitions, biomarkers is a measurable biological indicators of the presence severity or type of the disease in medical settings. Generally, clinicians use biomarkers for three main purposes. The first is for diagnosis. We can describe the observable characteristics of a certain disease. The second use of biomarker is for prognosis. Physician needs to do the risk stratification and decide the disposition of the patients. So we need to predict the patient's outcome and do some more aggressive intervention before the adverse outcome happened. And the third purpose for the biomarker is usually the treatment, to monitor the treatment response. And biomarkers may be a more sensitive marker than the clinical manifestations to inform the physician whether the initial treatment is effective or not. So let me summarize the current evidence on several clinically available biomarkers and their prognostic or diagnostic accuracy on COVID-19. Let's start with the most commonly measured biomarkers, white blood cell count. Increased leukocyte is universally observed in all series of studies that we can find patients with COVID-19 have elevated leukocyte count than patients without. However, there's lots of overlap. So the discrimination between severe or non-severe disease is not that good. However, we observe that the neutrophils increase and lymphocyte decrease. So we can use a neutrophil to lymphocyte ratio as a derived marker to observe whether and to select patients who may deteriorate. So in the retrospective studies, we found that the patient who deteriorate have a higher neutrophil to lymphocyte ratios than those non-severe cases. The average neutrophil to lymphocyte ratio of the severe cases is around 5.5, but for the non-severe cases, the NLR is around 3.2. However, other factors may disrupt the accuracy of the white blood cell count. Notably, glucocorticoid, such as dexamethasone, has been recently showed that they may improve the lung conditions in patients require ventilation or require oxygen therapy. And steroid itself will increase the white blood cell count. So white blood cell count itself is an initial check for the severity of the patient, but we need other more specific biomarkers to differentiate severe cases from non-severe cases. And platelet count is also an important marker. During the past 2 decades clinical observations, we now understand that platelet not only play an important role in coagulation, but also play a very, very important role in the immune response to the microorganisms. And a meta-analysis that consists of 700 patients revealed that severe COVID-19 patients actually have lower platelet counts than non-severe cases. And the non-survivors evidently have a significantly lower platelet count. Using thrombocytopenia as an endpoint also revealed a 5-fold greater risk of COVID-19. The odds ratio is around 5. So thrombocytopenia is an indicator of severe disease in COVID-19. And C-reactive protein, it is a very well-known, widely used biomarker that reflect the inflammatory conditions of the human bodies. It is produced by liver and many induced by the interleukin-6. So from the clinical observations, several series observed that for the severe cases or fatal cases, they have significantly higher levels of CRP than non-severe cases. And one study also analyzed the optimal cutoff, and it shows if a cutoff of a 41.8 milligram per liter, it is likely to progress to severe COVID-19 disease, which will be very, very useful for the frontline physicians, and that they can select some patients to admit to the ICU before they deteriorate. Overall, the area under the ROC curve for CRP to differentiate fatal and non-fatal cases is around 0.87, with a sensitivity of 83% and a specificity of 91%. And let's go to the interleukin 6. Interleukin 6 is in the upstream of CRP, and it can reflect the pro-inflammatory cytokine storm developed during the COVID-19. There is one meta-analysis that includes 6 studies. The mean IL-6 concentrations was 2.9-fold higher in patients with complicated COVID-19. The complication is defined as a patient who admitted to ICU, or a patient who developed ARDS, or a patient who died during the hospital course. And there's also another commonly used biomarker called procalcitonin. Procalcitonin was first developed as a tumor biomarker of thyroid cancer. However, the sensitivity and specificity was suboptimal, but accidentally they found that patients with fever, especially patients with severe bacterial infections, will have markedly elevated procalcitonin. Therefore, procalcitonin has been used clinically as a marker to differentiate bacteria from virus disease for about 2 decades. Recent several studies also observed, although PCT in patients with COVID-19 was not markedly elevated, but the gradient of the procalcitonin also reflect the severity of COVID-19. There is a meta-analysis that demonstrates increased PCT values are related to a nearly 5-fold higher risk of a severe SARS-CoV-2 infection. And let's go to a D-dimer. Actually, during the outbreak in Wuhan, D-dimer was the first biomarkers that has been mentioned that can reflect the outcome of the COVID-19 diseases. And later, we also observed COVID-19 may induce several thromboembolic complications, such as pulmonary embolism or deep vein thrombosis. So, and we also observed that nearly 90% of inpatients with pneumonias had increased coagulation activities. So there are several clinical studies showing that inpatients who admitted to ICU will have higher level of D-dimer than non-ICU patients. And D-dimer cutoff of 2.0 microgram per milliliters is associated with in-hospital mortality for COVID-19. And at a later stage, we also found severe patients may have a myocardial injury, and therefore the high sensitivity troponin has been measured in patients with COVID-19. And there is a small series study who found troponin is the most strong indicators of mortality. The odds ratio is as high as 80 for high sensitive troponin I. So actually the troponin is a very late marker when the patient develop multi-organ failures and severe myocardial injuries. So although it is associated with high risk of mortality, it is usually not an early indicator of severity in patients with COVID-19. And there are several biomarkers, but for clinicians, we want to ask which one has the highest discriminations between severe and non-severe cases. There is no comprehensive studies that made head-to-head comparison for the above mentioned biomarkers. Luckily, we have a small cohort that is cooperative research with the Chang hospitals in China. We have a cohort of 252 patients, and we categorize these patients into 4 severity classifications. That is mild, moderate, severe, and death. You can see that we compare the gradient of 5 markers. This is white blood cell count, lymphocyte, procalcitonin, CRP, and D-dimer. You can see that for WBC and lymphocyte, it just cannot reflect the 4 kind of different severity class. For procalcitonin and CRP, the gradient is good, and it's better than the D-dimer. And we further do a comparative ROC analysis. You can see that the CRP and the PCT are the 2 markers that has the highest ROC area under the ROC values. D-dimer and lymphocyte count have inferior area under the ROC curve than PCT and CRP. So we know that if we want to differentiate patient severe cases from non-severe cases, the initial choice may be the procalcitonin and the CRP. However, there are other considerations that clinicians may need more information to make clinical decision. One of difficult decision is whether we have to add empirical antibiotics for COVID-19 patients. Currently, the surge of COVID-19 patients could lead to a surge in antibiotic-resistant bacteria, because we know that around 8% of cases have super infection of a bacteria or fungus. There is no reliable diagnostic tool that the frontline physicians can differentiate patients with bacterial super infection than those with merely viral infections. So broadly spectrum antimicrobial therapy used as empirical combination therapy is a common practice in emergency department. However, if we use empirical antibiotics for all patients with COVID-19, then the next wave is severe antibiotic-resistant bacteria, another pandemics. Therefore, we first conducted an analysis. We analyzed our patients. We have an Italian and Chinese cohort. We combine them and do this analysis. We see whether patient received empirical antibiotics will have better outcome than those who did not receive empirical antibiotics at the emergency department. After very comprehensive propensity score matching analysis, we show that patients with empirical antibiotics did not have better outcome than patients who prescribed with broad-spectrum antibiotics in the emergency department. And so we know antibiotics is not a panacea. We need to select a very few patients. Actually, it's only around 10% of the patients that have evidence of bacterial superinfections. In this scenario, procalcitonin may be better than C-reactive protein. This is the pathophysiology of the procalcitonin secretions. Procalcitonin is induced by lipopolysaccharide, IL-1 beta, and TNF alpha, but it is inhibited by interferon gamma. So for the clinical observation, we see that a patient with systemic bacterial infections usually have higher procalcitonin levels than patients with viral infections. And in last year, we conduct studies that we want to combine the procalcitonin with a multiplex PCR to guide the antibiotic use in the emergency department. The multiplex PCR system we use is a film array system, which can test about 17 virus and 3 bacteria in 60 minutes. Currently, the film array panel also include the SARS-CoV-2, so they can give you the results of viral tests not only for the SARS-CoV-2 but other 17 common virus in the emergency department within 1 hour. The virus result is not sufficient to make a decision on the antibiotics use. So we also combine the information of procalcitonin. We give the physicians the result of a procalcitonin with the film array RP result. Then it shows that the physicians with the viral test results and the procalcitonin results are more willing to withhold the antibiotics. You can see that the duration of intravenous antibiotics in physicians with the information of a procalcitonin and the multiplex PCR of virus have 4.4 days less of intravenous antibiotic use. And also, the physicians with these information are more willing to stop or de-escalate antibiotics. So, although procalcitonin and C-reactive protein have a very similar discriminative capability for severe and non-severe cases, procalcitonin has an advantage of differentiate bacteria from viral diseases. So if you want to make a decision on the use of empirical antibiotics, I think procalcitonin may give you more information. And there's another thing we need to consider, which is the dynamics of these biomarkers. During the sepsis, there are 3 main types of biomarkers that will rise in different time frame. The IL-6 and the IL-10 or TNF-alpha rises first, but their half-life is very short, so they will soon go down. The procalcitonin rise around 6 to 24 hours, and the CRP will not rise until the 24 to 72 hours. So the physician must determine which marker you want to measure based on the presentation time of the patients. If the patient presents to the emergency department earlier, you probably have to select the marker that rises early. If the patient presents late, then you probably select that the markers that elevated late. So these are dynamics, not only for the accuracy that the physician needs to select their biomarkers for the patients. And for the procalcitonin, in August, there's a new publications. You can see not only for the initial measurement, you can see that the discharge cases mean the patients recover from the COVID-19 have the procalcitonin is high on presentation, but after discharge, the procalcitonin was decreased to the normal levels. So we can use the daily measurement to observe the course of the COVID-19 for the patients. And you can see that for the death cases, the procalcitonin will elevate during the hospital courses. So the dynamic monitoring of the biomarkers may provide more prognostic information than your single point measurement of the biomarkers. And if you compare the IL-6 and the procalcitonin, the IL-6 have even shorter half-life. So if you want to measure the improvement of a certain treatment, less than 1 day, for example, then a better choice may be IL-6 because the half-life of IL-6 is shorter than the procalcitonin. And we know that neonate is a very special group of patients for several biomarkers. For example, procalcitonin and the white blood cell count are highly elevated during the 48 hours after birth. These are called the physiologic elevations. So IL-6 have a very special advantage in the neonates. There are several analyses. All of them point that IL-6 is a better marker of systemic infection than the CRP or procalcitonin in this group of patients. I mean the neonates, especially during the 48 hours after birth. And there's also another study shows the combination with IL-6 and CRP can help diagnose neonatal sepsis. And you can also use the cord blood IL-6 to early detect whether the neonate have neonatal sepsis. We do not have the data of COVID-19 in neonates, but the past experience may inform the future researchers that IL-6 may be a promising marker for the neonate with COVID-19. And last one, I will present the two promising biomarkers for COVID-19. One is the mid-region pro-ADM. ADM, which is adrenal modulator, it is a multipotent regulatory peptide with a number of biological activities, including vasodilation, positive inotropic effect, diuretic, natriuretic, and bronchodilatic effect. It is upregulated by hypoxia, inflammatory cytokine, bacterial products, and shear stress. And ADM is even present in the pneumocyte type 2, smooth muscles. So it is a very promising molecule in the region, in the area of ARDS. Actually, for the past studies, pro-ADM proved to be the best biomarkers in community-acquired pneumonia to predict the short-term mortalities. And last month, there's a paper comes out that compare the pro-ADM with PCT, CRP, NT-proBNP, LDH, and D-dimer. As predicted, MR pro-ADM has the best discrimination compared with all other biomarkers that I mentioned. So this is a very promising biomarkers, and maybe they can implicate the basic scientist how pro-ADM will inform the grave prognosis of the COVID-19 patients, what pathway they may lead to, and you basic scientists can use the information to design new treatment. And the last one is the heparin binding protein. Heparin binding protein is a preformed protein in the neutrophils. The heparin binding protein can induce the capillary leakage. You can observe this fluorescent image of the capillary. After infusion of heparin binding protein, the capillary protein leakage to the surrounding tissues. And the use of heparin binding protein in sepsis has been 10 years. One of the most important characteristics of HBP is that it can it will be elevated 12 hours before the septic shock. So if we want to detect patients who will deteriorate in COVID-19, HBP is another choice. And this is the Linders group from Sweden. They first found that HBP have an important role in sepsis. Recently, they published the result of the observation of HBP in the COVID-19. They show that the RC curve is around 0.86. Although they did not do a head-to-head comparison with other biomarkers, but it is shown that the HBP were elevated 72 hours before the organ dysfunctions. So let me make the last slide of the take-home message. Biomarker may be an important diagnostic tool for risk stratification and monitoring disease course in COVID-19. CRP and procalcitonin, which is widely available, showed high predictability of in-hospital mortality. IL-6 may have a role in neonates for early monitoring disease progressions. PCT has another advantage is that it can help clinical decision on the empirical antibiotics. And emerging promising two biomarkers, MD pro ADM and HBP, may have a better performance than the above mentioned biomarkers. Thank you for your attention.
Prof. Lee, thank you very much for the sharing. Before we move to the next talk, I do have questions regarding the biomarkers. You have shared with us that the biomarker may be different in different groups, particularly the neonates and other groups. And we know that COVID-19 can cause a lot of severe effect outside of the lung. So there have been reports regarding kidney damage and also skin condition or even memory loss by different patients.
Yes.
Do you foresee that there will be other biomarker subsets that will be particularly developed for these different patient subgroups regarding the particular side effect they're suffering from?
Oh, yes, definitely. For example, a certain group of patients develop acute kidney injury, and there's a bunch of biomarkers that specifically to predict the acute kidney injury. For example, NGAL, LFAP are two of the most promising biomarkers. And also there are biomarkers from brain, from liver, from gut. So I think for the organ-specific biomarkers is an area that has not been fully developed. So the most of the biomarker I mentioned today is an overall mortality prediction biomarkers.
Thank you very much. And another question is, are all these biomarkers detectable at tissue level, or are they mainly blood-based biomarkers?
There are many blood-based. They are many measured from the our peripheral blood circulation. Yeah, because from the clinician's perspective, if you need to do the tissues, it cannot give you a timely information to make a quick decision in the emergency department. So the biomarker I mentioned from a clinician's perspective is from the peripheral blood. You can readily measure them.
Thank you, Prof. Lee. Thank you very much for answering the few questions we had. So now we will move to our next talk. So just now, Prof. Lee had mentioned about that a lot of the biomarkers that have been sharing in these sessions are mainly blood-based, but we do know that COVID-19 has been causing a lot of damage to the tissue. So sometimes tissue information or spatial information is also important for biomarker discovery. And this brings us to the next talk, to our next speaker, so Dr. Arutha Kulasinghe, who will share with us on how he used spatial biology to discover biomarkers at the tissue level. So Dr. Arutha Kulasinghe, yes, I can see your slides now.
Perfect. Lily, sorry, QQ, can you hear me?
Yes, I can hear you very well.
Perfect, great. Am I good to go?
Yes, please start your talk. Perfect. Thank you.
All right. Great, thanks, QQ. I'd like to just thank Prof. Ruby for this invite, and it's great that NanoString is, doing these seminars. I think it's really timely, and yeah, I was really fascinated by Prof. Lee's talk, and I think I'll probably reach out to him after this with some of the basic work that we've done in the lab, and we're keen to share. So I'm based in Brisbane, sunny Brisbane, in Australia, and this project, or the title of this work's called Spatial Profiling of Lung SARS-CoV-2, and I can't see the top there, and that it dissects virus-specific host responses and gene signatures. So my background is I'm a cancer biologist by training, and we've been working with nanostring over the last couple of years, and we really got excited by this technology for its application in the immuno-oncology space, because you could essentially tease apart the tumor microenvironment and then understand the tumor and immune cells and the context within a tumor. And how this is done and how this has led on to COVID is you're able to really, the beauty of this technology is that you can use archival tissues. So, you could probably go back a couple of years and everyone's got access to these archival tissues. And serendipitously for us, we had collaborators in Iran, Chile, and Brazil that had COVID-19 rapid autopsy samples. And these were FFPE blocks at the time. So we had them sectioned onto glass slides. And what you're able to do with a pathologist is to define these tissue sections and identify different regions of interest. So you can look at about 100 proteins or up to 20,000 RNA targets in a single tissue slide. So I think that's one of the beauties of this technology, and you're essentially able to get digital expression data, be that protein or RNA, for your tissue. So this paper came out in May of this year, and it was a really important paper in the field in that it ran a nanostring bulk gene expression analysis of rapid autopsy samples from COVID patients and compared it to that of H1N1. And this sort of started things moving along in my head, where we thought, what about the spatial aspect of it? And they showed in this Venn diagram, there were genes associated with COVID exclusively. There were those associated with influenza exclusively, and they were shared, obviously, in that study. And this was from a known sort of a panel of nanostring panel that was run on the sample set. So what we thought we'd do was to look at the spatial data set if we could, and we reached out to collaborators in Brazil where they had rapid autopsy samples from COVID patients, pandemic 2009, pandemic H1N1, and normal healthy non-viral tissue as well. So the workflow for us was, this work initially began in lung autopsy samples, but we've expanded it to myocardial, and we've actually got placental material as well, which we're hoping to do in the near future. We've got the myocardial data back, but the placental work is looking at vertical transmission of the virus late in pregnancies, and that's some work we're hoping to do with Professor Huang. And then we do H&E staining, Mason's trichome to identify the fibrosis in the tissue, IHC for the spike protein, RNA scope, which is essentially RNA-ish for SARS-CoV-2, TMPRSS2, and ACE2. We do path review. So for cancer, it's pretty straightforward, but for COVID, it's very complex. So initially, when we were looking for a pathologist to look at this, the tissue sections, I couldn't find anyone in Brisbane that had seen COVID-19 lungs. So this was a problem, because obviously in Australia, we're pretty lucky, and in Brisbane, we're pretty lucky that the caseload wasn't too high. So eventually we were able to reach out to a lung pathologist who came on board and was able to really nut down and look at the tissue, the structures, the cell types, and identify where the virus was binding to. And then we ran the nanostring geomics DSP COVID-19 Immune Atlas panel, which is about 1900 genes. So it's based off the cancer transcriptome CTA Atlas panel with a number of COVID spiking genes. So it's got a good representation of immune and non-immune genes in that panel, and then obviously the bioinformatics with spatial data sets. So the tissue was, there were blocks, there were tissue microarrays. So we had 10 COVID cores. Each core represents an individual individual patient, sorry, and then pandemic H1N1 in the middle, and then the normal lungs. So this is taken so this is just Mason's trichome looking at the fibrosis. And so this is in one case where we can see, so this is the RNA scope data. what we can see in the red is the virus. And in this course, so what was really interesting in the study was that we had patients where there was high RNA scope, the red signal was really high in them, but at the same time, in some of the patients, in about 8 out of the 10, they were really low. So this, you know, the RNA scope was almost at background level. So I think at that time, that's when we were able to stratify, okay, we've got high virus versus low virus here, and that matched up with our IHC spike data. And just on the right here, this is what the nanostring DSP multiplex IF looks like, stained for pancytokeratin, CD68 and CD3 and DAPI. So we were able to then, so this is just in my office, and we actually needed a fourth screen, which we did get in the end. So we had the nanostring image on the left, the multiplex IF. In the middle screen was your spike protein, on the right was your H&E, and then we had the RNA scope on the fourth monitor. I wasn't able to get a photo for that. But what we were able to do is to identify the cell types within the regions of interest. So we knew where the hyaline membranes were in the lungs, the type 2 pneumocytes. And the beauty of the technology is, if you look at the bottom image here, this is a bronchoepithelial section where we're able to see in the, so this is multiplex IF on the left, and then going to the right, you've got your IHC for the spike protein, so you can see where the brown staining is. Then you've got the H&E and the Mason's trichome going to the extreme right. So what you're able to do is do custom regions of interest, or draw around these structures where you can see there is, and we had the matching RNA scope image as well to see that where the virus is binding to, and then we could liberate those 1900 plex mRNA targets for those regions of interest which we defined. So this is what it looks like on the instrument. So on the first slot is your COVID sample, in the middle is the H1N1, and the third is the normal. So the two cores which had really high virus were really well sampled, and we had about 1 ROI per core, and then across the H1N1 and the normal healthy. So this is what it looks like when you're doing the region of interest selection on the instrument. So the gridded, so the shaded area is where the signal is liberated from. And if you look at region 12, 38, and 39, what we've tried to do here, and what will get, I think, better with time, is that you can start to grid away and look at gene expression through the not the microenvironment, but through the tissue structures in here. And I think this is a really powerful technology that we can, you know, really harness for COVID-19, but also for other applications too. And so this is just a snapshot of what it looks like when you're doing it live. Someone told me that that's me taking a photo of it in the background, probably early in the morning. So you're able to really grid. So if you can see this white outline, that's you, that's me actually going through and choosing this bronchiole to liberate the signal, and then that will be assessed for about 1900 markers spatially. The cytokeratins in green, the CD3s in red, CD68s in yellow, and DAPI. So these are your morphology markers, and then you get the transcript level data afterwards. So just diving into the data here. So if you ignore the left-hand image, which is a bit busy, so this is a PCA plot or dimension reduction analysis of the transcript level data. And what we see here is that COVID in the blue and H1N1 in the red, and the normal healthy in the yellow. So what we see at first glance based on PC1 and PC2 is that the COVID and H1N1 is separate from normal, which is great, which is reaffirming. And then there is overlap between H1N1 and that of COVID, as expected, and it was reported in the New England paper as well. So that was reaffirming to us, and we knew that there would be overlap between H1N1 and COVID. These are, you know, they're autopsy samples. It's severe COVID versus severe H1N1. Then when we separate it out based on cause, so the cause represent individual patients, we can see a separation of the data. So you can see that there's a population here at the bottom that, which we couldn't explain. So you're looking at, it's almost like a helicopter overview of your transcript level 1900 plex data set. And so this is by patient sample, but what got really interesting was we couldn't really explain what this population was at the bottom. And then when we overlaid our transcript data with our pathology assessment, now you can see where the transcript data is coming from. So you can see your bronchoepithelial cells are at the bottom here. The light blues, your type 2 pneumocytes. Your macrophages are in the yellow and in the dark brown. Your lymphocytes are in the dark green, and so on. So I think this is where it becomes really interesting, where you can overlay your RNA data with your pathology data. And that was really informative, because we could identify straight away where the bronchoepithelial cells and that signal was coming from. So just looking at some differential expression analysis. So I think I'll have to probably reach out to Prof. Lee about this later, but this is between COVID-19 versus control. And one of the top hits that was differentially expressed between COVID versus control was, so you had a number of different sort of immune-related genes at the bottom here, but your type 1 interferon response were at the top. So IFI27 and Ly6E were two of the top hits. So as I said, my background's in cancer biology, and luckily we had a virologist on the call, and she just lit up when she saw this, because IFI27 is known to differentiate between viral and bacterial pneumonia, and it's known to be upregulated in nasopharyngeal samples and in peripheral blood samples of COVID patients. But the data wasn't there till recently to show that this, this was actually, the signal was actually coming from the tissue. So we're going into this at a greater depth, and we've actually run a number of serum samples, and we can see that IFI27 sort of stratifies with disease severity. So in moderate and severe cases, it's heavily elevated, and then it stratifies back down. So we're looking at this at a greater depth, and it was a really interesting finding from this data set, which we're looking to pursue. And I'd be interested to talk to Prof. Lee about adding this onto his panel, actually. So we're working with a few researchers from Sydney to develop this further. And there were other sort of fibrosis epithelial cell markers in that data set. And when we look at, again, so this is between COVID and H1N1, and it was interesting, again, severe COVID versus severe H1N1, there weren't many genes that were differentially expressed. And again, IFI27 was up there, IFI6 too. So we're looking at developing this as a potential combination biomarker to stratify disease for COVID. So I think that'll be really interesting that IFI came up again, and some of Ben Tang's work from the government and UNSW has supporting data for peripheral blood and nasopharyngeal samples. So we're really excited by this finding. And then we looked at virus high versus virus low. So when you say by cause, that's pretty much by patient samples. So you can see your type 1 interferon response genes are up again. And if you go to the right here, this is viral load high versus viral load low by region of interest. So this is taking your total 60 regions across the samples and measuring it against your viral load. So it's putting all the COVID samples together and measuring. And again, the type 1 interferon 1 responses there, and a few other markers of interest. And then we did some gene enrichment analysis, and this was pretty interesting in that we were able to identify, again, your interferon gamma response genes were enriched for your blood coagulation, your angiogenesis, your interferon alpha response, and hypoxia, and the matching waterfall plots on the right here. So you can look at the different pathways that are activated between COVID versus control. We have done the analysis between COVID and H1N1, and that should be in the paper. And with that, I'd like to just thank this. It's been an incredible effort. Again, coming from a tumor biology background, without the virologists, without the immunologists, without the clinicians, the study couldn't have been possible. And we're really excited by this initial data set. And we've actually just got back the myocardial data, and we have matching samples from the lung, of the lungs from some of the patients. So that's really exciting for us. And we're hoping to look at that at a greater depth and placental material in the future too. And please go and have a look at this paper. It's on medRxiv. It was indexed over the weekend, and we will be, hopefully we made a submission to a journal as well, and that'll come out in print too. Thank you for listening to me, and thank you, Ruby and QQ and the team.
Thank you, Arutha. Thank you very much for the great sharing. It's really amazing to see how the cell type in spatial and also the pathology perspective can actually match back into at the transcript level. Really some incredible information that we had never seen before. We do have a couple of questions from the audience. So There's one question regarding the genes being upregulated in the tissues. So the question is, why are the cytokeratins being upregulated, and are they related to any particular pathology of the tissue?
Yeah, that's a good question. So this panel, QQ, it's based off the CTA, so the Cancer Transcriptome Atlas. So it's not an unbiased approach. So these were the findings that we found in the data set. Obviously, a whole transcriptome approach will tease apart the data a bit more. Yeah, it's a good question. We haven't really thought of that, but I think it probably, it's probably to do with epithelial cells and that sort of component in the tissue that's being picked up there. And one of the cases was from a patient that died of carcinoma. So it is a mixed cohort. They're non-viral though, but yeah, we are looking at it at a greater depth now.
Thank you very much for the sharing. And I do have one question, and maybe this is also related to Prof. Lee's sessions. So for Prof. Lee's session, the biomarker discovery really is focusing on the blood-based biomarkers, and while your sessions is really focusing on the postmortem samples, basically it's when the patients are deceased, and then we will be able to analyze this marker. Do you foresee some middle ground where like we can monitor the patient's progress after admission of these COVID patients? Because this is where the patient's disease start to change into mild disease, severe disease on those who actually need ICU attentions. So this is where we see the medical resource become exhausted. Do you see any way that we can combine the tissue-based biomarkers and also the blood-based biomarker from Prof. Lee's session, which we can use as a monitoring tool for the COVID pandemic?
Yeah, absolutely. I think, the peripheral blood nasopharyngeal samples are, those are the most accessible sample type. Obviously, the tissue data is important in teasing apart, empirically where it's coming from. And one of the findings, just IFI27 that we were able to show in our data set, it was reassuring that was elevated in the peripheral blood of patients. We've gone back and looked at day 0 serum samples and from mild, moderate, and severe cases, and we can see that it's elevated in the moderate and severe, and it's not there in the healthy population. So we are, ultimately it has to be in a non-invasive sort of manner where you'd be able to sort of triage these patients or have a triage biomarker of sorts. If you're able to assess at day 0 whether these patients would progress, to being on a ventilator or not, then that's really useful, especially where in the Northern Hemisphere where you have sort of dual influenza and SARS-CoV-2 infections, and there's a backlog on COVID testing. I think biomarkers, and especially peripheral blood biomarkers or nasopharyngeal swabs, urine, are really useful, but I think it needs to be based in the tissue data and findings from the tissue data.
Thank you very much for the sharing. So one question from the audience is that whether this RNA profiling analysis is for whole transcriptome or is this for targeted approach?
Yeah, so this was from the targeted approach. So it's a 1900 plex RNA panel. We are hoping to run a whole transcriptome on these samples in the near future.
Thank you very much. And again, we had actually came to the end of our sessions for today's webinar. And we want to thank Prof. Lee and also Dr. Arutha Kulasinghe for their great sharing on the biomarker discovery, both in blood and also in tissue. And on top of that, I'm also going to share with the audience the paper that Dr. Kulasinghe just shared in his research. So I will put that into the chat, and for those who are interested, please feel free to take a look. And also actually after the biomarker discovery journeys that we discussed today, it will bring us to the next session, which is going to happen on 17th of November regarding the disease dynamic and the treatment. So just now we have seen that the biomarker discovery can be either at the patient admission or at the postmortem level, but we also want to know how the disease dynamic, how the COVID-19 is going to change the host response, and how doctors can develop new treatments and therapies to help the patients. So I will put the next link of the webinar in the chat. Please register if you are interested into looking into the disease dynamic. And again, we want to thank our speakers and also our organizer, Prof. Ruby Huang, for today's webinar organization. So Prof. Huang, I would like to know if you have any closing remarks for today's sessions.
Thank you everyone for participating. I think this webinar from Biomarker, we have a journey from the blood, from serum, and then to the postmortem tissue. I think marry these two together, then we could have a good comprehensive view of what COVID-19, this disease, is really about, what the virus is really doing for the patients. So I think this is very exciting, and I hope that Arutha and Prof. Lee, you guys can, reach out to each other for more collaborations. And then so are for the audiences, if you have any ideas that you want to collaborate with Prof. Lee or with Arutha, please reach out. Thank you very much.
Thank you, Prof. Huang. And again, Prof. Huang just mentioned that there are lots of collaborations that we have seen in these webinars. So for those who like to look into a COVID-19 research collaboration, please feel free to reach out to me and to any of the speakers. This webinar series is also to promote the COVID-19 research collaboration across Asia Pacific. With that, I am going to leave the webinar room open for another 5 minutes. And I had input the link, the paper link by Dr. Kulasinghe's paper in the chat. Please feel free to take a look at it. And I'm also going to put in the webinar's link for our next sessions. So thank you, everybody. I will see you next week.